Hi everyone, it's Dr. LeBlanc with Unit 8, Recombination and Transposition. Our first topic is homologous recombination. Our learning goals are to understand the Holliday model of homologous recombination, to understand the double-strand break repair models for homologous recombination during DNA repair and during homologous recombination in meiosis, to learn how to interpret electron micrographs of chromosomes crossing over during meiosis, to understand the role of homologous recombination during gene conversion via mismatch repair and DNA gap repair synthesis. This is a transmission electron micrograph of homologous chromosomes crossing over during meiosis. This structure right here that forms an X between the two chromosomes that are crossing over is called a chiasma. This is an artist interpretation of the actual electron micrograph. Homologous recombination occurs in all species by several different mechanisms. It's best understood in E. coli. Even though E. coli is haploid, a cell can have more than one copy of the chromosome during replication before it divides. In eukaryotes, it's most frequently seen in meiosis, but it can occur in mitosis as well. This is a diagram of crossing over between sister chromatids. It's also referred to as sister chromatid exchange. Here we have a single chromosome, one chromosome. You can tell it's just one chromosome because it has one centromere here. It is comprised of two different sister chromatids. If you get an exchange between two sister chromatids, you are not changing the genetic material. This chromosome has dominant alleles at the A gene and the B gene, and it's copied during DNA replication and you make another sister chromatid. So crossing over does not produce a recombinant chromosome. However, during meiosis, you get crossover that occurs and the formation of recombinant chromosomes. This occurs in meiosis 1. Here, meiosis 1. Okay, so here you have homologous chromosomes. These are same chromosomes. Let's call them chromosome number 3. This chromosome number 3 you got from mom, and this chromosome 3 you got from dad. So happens that the chromosome you got from mom has the dominant alleles for the A gene, and the one you got from dad has the recessive alleles from the A gene. And the ones, again, for the B gene, you got the dominant allele from mom and the recessive gene from dad. If there's a crossover right here between homologous chromatids, okay, you will end up with recombinant chromosomes. At meiosis 2, during meiosis 2, the centromeres split apart, and each of these new single monovalent chromosomes is going to go into a new cell. This is non-sister chromatid exchange. That goes into its own cell. So each of these cells could be a sperm cell. And this chromosome went into this cell, this chromosome went down here, this chromosome went to here, and this chromosome went here. So you end up generating 4 different sperm cells, each with a different combination of alleles for the A and B gene. This chromosome has the 2 dominant alleles. This has a dominant A, recessive B, 2 recessive alleles, recessive A, and dominant B. The exchange of DNA between non-sister chromatids of homologous chromosomes yields genetic recombination. The Holliday model for homologous recombination accounts for the major broad strokes of what we see in recombination in meiosis. The evidence that supports this model we saw in electron micrographs, and here's some more electron micrographs. And we see again the or chiasma here, right here. There's a crossover event here. There's also a little video about the Holliday model. These are the broad strokes of the Holliday model. Here you have homologous chromosomes. And if we look very up close and personal at this particular region right here, you can see that these two chromosomes have to line up very close together. Now, the Holliday model proposes that there are nicks that occur in one strand of each of the homologous chromosomes. This is the part that's probably not what happens exactly this way during any of the homologous recombination repair mechanisms or exchange in meiosis. Okay, the two chromosomes definitely align very tightly together, and there is nicking that goes on, but it's probably not in this configuration. The Holliday model then proposes that you have strand exchange and you have this chi structure or the Holliday junction that can then migrate. This is called branch migration. And as it migrates along the chromosomes, you flip around the different strands of DNA between the two different homologs. The heteroduplex region, it may have some mismatches. That's why they do it with this little squiggly line here. Strand invasion is something that we saw from DNA repair mechanisms. And the Holliday junction is capable of migrating laterally. So here we've seen it migrate in this direction here. It's also called branch migration. After the branch migration is completed, and you need to resolve the junction, in other words, break it. You can break it in two different geometries. If you do it this way, you'll end up with new chromatids that have not exchanged a substantial amount of material, just a little bit here at the Holliday junction. If you cut it in another configuration, however, you can end up with recombinant segments of the chromosomes. This is referred to as isomerization and resolution, and the only difference between the two chromosomes is this region that exchanged. Now, we have to refine the Holliday model. This is another chi form of the chromosomes demonstrating some evidence in support of the Holliday model. The Holliday model accounts for the general properties of recombination by the chromosomes in meiosis. However, it's more likely for one DNA helix, not both of them, but just one of the double-stranded DNA molecules to incur a single nick or a break in both strands, thus double-stranded break repair. What we're going to see in the next couple of slides is unmatched DNA repaired by DNA gap repair synthesis. Okay, so this is what really happens. You get a double-stranded break. This is exactly what we've seen with the double-stranded break repair that involved the BRCA genes, for example. Okay, so you get a double-stranded break repair either on purpose or a spontaneous catastrophic event. You get processing so that you end up with single-stranded 3' ends. Remember, this is 5', 3', 3', 5'. So if this is the 5' end here, this is the 3' end right here. Okay. This 3' end will get coated with Rad51 or Rec-A, for example, from E. coli, and then invade the other strand, and you create this D or displacement loop. Since this is the 3' end down here, we're going to synthesize some DNA from here. We'll also synthesize some DNA over here. So here we are again. Here's our D loop. Here's our 3' end that we're going to synthesize some DNA from. The gap repair synthesis fills in the vacant regions. This is DNA synthesis right here. So here's our 3' end up here as well. So we're going to fill in the DNA up here. That's the gap. Okay. We don't show the formation of the heteroduplex DNA here for simplicity, But the DNA gap repair synthesis fills in the short region where the DNA is missing, and you can get some branch migration in either direction and resolution. And you can either end up with non-recombinant chromosomes or recombinant chromosomes. This is at the crossover site. Notice that you've got, in this case, you've got purple chromosome here, purple chromosome here. So this original all purple chromosome is now exchanged a good portion of it with the blue chromosome and vice versa. It's a reciprocal cross. During meiosis 1, we actually want to create double-stranded breaks to perform recombination or crossing over. SPO11 is a protein that makes the first double-stranded break. So there's actually a protein that breaks the DNA. It is in an very controlled manner because these two sister chromosomes are very, very tightly aligned with each other already, even before the break occurs. The first meiotic division, meiosis 1, requires the formation of crossovers between homologues by meiotic recombination. And SPO11 is the protein that makes the first cut. And then after that, you have RAD51 coming in and encoding the single-stranded region. And it also helps this DNA to find its precise, align precisely with its homologous sequence here so that you have homologous base pairing, A to T, G to C. And then we can synthesize some more DNA in this direction using this as template right down here. And then by one of two mechanisms, this is resolved. This is what usually happens in meiosis because it ends up resulting in a crossover. There are two pathways by which this can be resolved. You've got the double-stranded break repair pathway using, for example, the BRCA genes and RAD51, or you can have the synthesis-dependent strand annealing pathway, this pathway. But this is the one that's the most common, the desired outcome for meiosis, what occurs in meiosis 1. Okay, so a lot of proteins that we know of that assist in homologous recombination come from our knowledge of what happens in E. coli. In E. coli, there's a protein called REC-A, and let's see if we can figure out which eukaryotic protein it's homologous to. REC-A binds to single-stranded DNA and promotes strand invasion. Ooh, we know a protein that does that. And it enables homologous strand to find each other. It also promotes displacement of the complementary strand to generate the D loop. So REC-A in E. coli has an equivalent in eukaryotes, and that equivalent is RAD51. Of course, we know there are a lot of other proteins too, but this is the one that I just wanted to pay attention to now. Gene conversion. What is gene conversion? Gene conversion is the homologous recombination where two different alleles become identical. Okay, so there are two different alleles in a nucleus on your two copies of your chromosome, and all of a sudden they both become the same allele. That is what gene conversion is. Gene conversion can occur in one of two ways: DNA mismatch repair and DNA gap repair synthesis. This is the model for DNA mismatch repair. So let's take a close look at our two sequences here. They're from the same regions of two homologous chromosomes in the cell, and they have nearly identical sequences here, except that on one of our copies, one allele has a GC base pair and the other allele has an AT base pair. Okay? The branch migration is initiated, and it starts to travel past this region where we have a difference in alleles, in the sequences of the alleles in the two different chromosomes. As the branch migration travels over this region, we're going to flip this DNA now up to a hydrogen bond to this DNA here. And when that happens, we've created a mismatch. Okay, so now this T is trying to base pair with this G up here. Here's the T trying to base pair with the G. Okay, we know that's not a correct base matching. And then this A is going to try and base pair with this C. So that's a mismatch. Okay, when the mismatch is resolved, it can be resolved in 4 different possible combinations. 2 of them convert the gene to one of the 2 alleles that existed at the beginning. So if we have a gene conversion, here we have both of these base pairs now being a GC base pair, whereas up here we had allelic variation. We had one allele with a GC base pair and the other with the AT. So we've converted, in this case, both alleles to the GC. We could also convert both alleles to the AT base pair. There's also a possibility that we just simply restore the allelic variation that we saw in the beginning, and that one chromosome has the GC base pair and the other one has the AT. Okay, so we discussed gene conversion by gap repair synthesis. We discussed it previously, but we did not pay attention to the possibility that there were differences in alleles. So if you have one allele on one chromosome and the other allele on the other chromosome, it's possible that after the gap repair synthesis, you end up with the same exact allele on both chromosomes. Okay. Basically, you have removed this allele by processing. Okay? The region adjacent to the double-stranded break is digested away, which eliminates the recessive B allele. So the only information you have left is the dominant allele from the other chromosome. And that is a second way to convert one allele to another. So in summary, homologous recombination occurs in all species. The Holliday model is based on the supposition that one strand of the double helix is broken at the exact same spot on homologous chromosomes. That's not really likely. Instead, it's more likely that a single double helix, will incur a double-stranded break. After the break, processing of the ends, strand invasion, the formation of a D-loop, DNA synthesis, and resolution occurs. RAD51 or REC-A coats the processed single-stranded ends and accomplishes the strand invasion. Homologous recombination is the mechanism by which recombination during meiosis I takes place. The protein that makes the double-stranded cut in meiosis is SPO11. All organisms are capable of undergoing homologous recombination, and even E. coli, though haploid, can harbor 2 copies of its genome after replication before the cell divides. Homologous recombination in E. coli uses several proteins that have homologs in eukaryotes. REC-A is the homolog of RAD51. Gene conversion is the phenomenon of 2 different alleles becoming identical using the homologous chromosome as a source of sequence information. And finally, homologous recombination can accomplish gene conversion by 2 different mechanisms: mismatch repair and gap repair.